Hello. Very good evening to the student who have joined. Can you please let me know if my voice is audible to you? In the chat box, please let me know if my voice is audible and if you can see the video. Okay. Thank you so much for confirming. Now, please let me know if you can see the presentation. Let me know if you can see the PowerPoint presentation. Okay. Thank you so much for confirming. We will wait till it's 6 p.m. and let more students join. Then we will start today's session. Meanwhile, please stay on the call. We will wait till it is 6 p.m. and we will start our session. So please stay. We will wait for 5 more minutes or 4 to 5 minutes, then we will start the session. Very good evening to the students who have joined. Please wait till it's 6 p.m. Then we will start today's session. Meanwhile, let me know if everything is fine with the audio, with the video, and also with the presentation. If there is any issue, please let me know through the chat box. We will start shortly, so kindly wait. While we are waiting for the other students join, if there is any doubt from the previous lecture, you can also ask in this time, right? If you have any doubt or any query regarding the last class, please you can ask it now. I think it's yeah, it's 6 p.m. and so let me start with the session. Okay, let other students join in the time. Meanwhile. Okay, so I hope my voice is audible and the presentation is visible to you clearly. If not, please let me know. So, very good evening to the students who have joined. My name is Mousumi Deb and I'm a PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAYAM course of Organic Chemistry in Biology and Drug Development, and today it is our week 5 live session. So what we do in every live session, so basically in each live session we solve that particular week's course content that has been released previously. So previously in this earlier this week, the week 5 course content was released along with the particular assignment for that particular course content. So what we will do in today's live session, we will solve MCQ questions related to the particular week's content. Like today we will solve questions related to the week 5 course content, and then every question will have 4 different options, and you have to let me know which of the following option is the correct in the through the chat box, and after that I will discuss that particular question and topic in detail. Before starting the session, let me clarify a few things. Like there will be a problem solving session every week. Okay, the problem solving sessions are scheduled on every Friday from 6 to 8 p.m. and the sessions are live on Google Meet platform. This is the link for every session. Okay, this link is common for every Friday class, so you can just write it down or copy it from somewhere and use the same link to join every class. Okay, if any network disruption happens during my class from my side, do not leave the session immediately. Okay, I will try to join as soon as possible, but do not leave the call. And if there is any issue from your net end and from your end, this call is open, so you can rejoin at any time. So you don't have to worry about it. Okay, I am recording the session. That is why I have muted your microphones and camera so that I can record the session without any hassle. And this recording will be shared to you after the session. Where to find this recording and this PPT, I will show you shortly. Okay, so during the session, you can post your doubts, your queries, everything through the chat box. Okay, although the microphone is off, although your microphones and cameras are off, I am monitoring everything here, and I will be communicating through the chat box. So if you have any doubt or any query, you can please let me know through the chat box. Okay, you can use the chat box also for answering in during the class. Okay, so you can answer during the class using the chat box, and also you can ask your doubts or your queries. Okay, since I'm recording the session so that I can record it without any problem, I have muted your microphones, and also I have muted your cameras. After the class, if the doubt still persists, you can drop an email to me. I will share my email ID at the end of the session. Okay, this presentation and class recording, it will be shared to you after the class. So where you have to where you can find this particular PPT and also this class recording, so you have to go to the SWAYAM portal. Okay, first you have to go to the SWAYAM portal, the announcement page. This is the thing you always you get through the email also about this announcement and everything. So what you have to do is, after going to this announcement section, you will find there is a left panel, okay, where the weekly course contents are released. Okay, and there you will also find your assignment which you have to answer, and at the end you will find there is something called problem solving session. If you go to it, you will find the problem solving session recording. Once you are clicking on the this problem solving session recording, it will redirect to you one page from where you can find a particular dates recording. You can see the recording through the YouTube ID, YouTube link, and you will find the PPT which is uploaded to a Google Drive. Okay, you will find a Google Drive which looks like this. It will have all the course content which is released. You can download and you can read. Okay, so lastly, we have for already we have 4 live sessions, so four PPTs are there, and the PPT which I am going to teach you today, that I will be uploading after the session. Okay. So again, I am saying if you have any doubt or any query, please feel free and write it in the chat box. Okay. I will answer your queries within the class. So let me just show you what are the course content, what are the today's course content. So as I have mentioned, today it is a week 5 live session. So what we do in every session, we solve MCQ questions from that particular week's course content. Okay, what is the week 5 course content? Then let us see. The week 5 course contents are like this. We have synthetic biology. So in week 4, what we had did, we have discussed about the our enzyme inhibition. We particularly focused on the enzyme inhibition, different kind of enzyme inhibition, like competitive, non-competitive, uncompetitive inhibition, suicide inhibition. We discussed what are the patterns of the inhibitions, what are the examples with the kinetics, how the graphs look like, what that we have discussed in our previous class. We also have solved couple of mathematical questions in our last class. There was few topics from synthetic biology in our last class also, like polyclonal antibodies, monoclonal antibodies. We had discussed about T cell, B cell, antigen antibody, and so on. So these things we have discussed already. So today we will discuss a little bit about the synthetic biology. Then we will move forward to the nucleic acid, and we will discuss about the DNA sequencing method. You have already know how to sequence the peptide chain, the amino acid sequencing. Today we will discuss a little bit about the DNA sequencing method, how to identify what are the particular bases that are present in a particular DNA chain, right? These things we will discussing today. So today I will share with you around 20 MCQ questions. Each question will be for we're having 4 different options. Okay, you have to identify after showing a particular question. I will wait for 2 to 3 minutes, and you have to identify A, B, or C, or D among 4 different options. Which of the following will be the correct option? And then you have to let me know it in the chat box. Okay, answering during the question, during the problem solving session, whatever I am showing you today, the question, answer, and everything, there is no marking, so you don't have to worry about that your answer is wrong or there will be a negative marking and so on. So it is not the thing. Okay, you can answer freely. Even if your answer is wrong, it does not matter. I will discuss it in detail, so you can check what is the correct answer. So let us start with the first question of today's session. So the first question of today's session is, in a particular human DNA, it has been observed that adenine is present in 30%. What will be the percentage of cytosine in the DNA sample? Your options are number A 30%, number B 25%, number C 70%, and number D 20%. Again, I will repeat. In a particular human DNA, it has been observed that adenine is present in 30%. What will be the percentage of cytosine in that particular DNA sample? 30%, 25%, 70%, or 20%? Let me know in the chat box A, B, C, or D which of the following you think is the correct answer of the problem number 1. I have already told you that even if your answer is wrong, it does not matter, but please try to answer. In a particular human DNA, it has been observed that adenine is present in 30%. What will be the percentage of cytosine in that DNA sample? 30%, 25%, 70%, or 20%? I will wait for 2 to 3 minutes, then I will check the chat box and we will discuss it. In a particular human DNA, it has been observed adenine is present in 30%. What will be the percentage of cytosine? It is very easy and I know all of you can answer it. Let me check the chat box. Okay, I got only one answer which is saying it is number D. Okay, let me check if number D is the correct answer or not. So, in a particular human DNA, if the percentage of adenine is 30, then what is the percentage of cytosine? The correct answer will be, very good, it is number D, it is 20%. Okay. So the rule of base pairing. So this thing is very important. The rule of base pairing in case of our DNA and RNA. Okay, so we all know there are 4 different bases. Actually, not 4. There are 5 different nucleic bases. that are the cytosine, guanine, adenine, thymine, and uracil. In DNA, cytosine, guanine, adenine, and thymine are present, and in RNA, cytosine, guanine, adenine, and uracil are present. Okay. So in case of the DNA, cytosine pairs up with guanine, adenine pairs up with thymine, whereas in RNA, cytosine pairs up with guanine, but adenine is pairs up with uracil. The thymine is not present in RNA; present in only DNA. Okay, so adenine makes a hydrogen bonds with thymine. Says 30%. So this is a very important thing that you have to remember, that adenine makes hydrogen bond with thymine. So since 30% of adenine is present and adenine is bonded with thymine, it means that there will be the amount of, or the percentage of thymine will be also 30%, since both are present equally. Okay. And also you have to remember one thing, I will show you again later also, that adenine and thymine, they are actually bound with two hydrogen bond. Okay, there are two hydrogen bond. Okay, let me just erase this. Between adenine and between thymine, there are three hydrogen bonds. Okay, so there are two hydrogen bond between adenine and thymine, and between guanine and cytosine we have three hydrogen bond. So this thing is you have to remember. Okay, so adenine is bond with, since 30% of adenine is present and adenine is bond with thymine, so means the percentage of thymine is also 30. Now, cytosine and guanine, these two are also present in an equal amount. Okay, so what is the remaining percentage of total cytosine and guanine? It is 100% minus 30 plus 30 means 40%, right? Because this particular, since adenine is present 30%, thymine is also present 30%, total it is making 60%. So the remaining two bases, cytosine and guanine, consist of total 40%. Now cytosine and guanine, they make hydrogen bond with each other, which indicates that they are present in the equal amount. Hence the percentage of cytosine will be 40 divided by 2, which is 20%. Okay. So that is how you have to solve this question. It is very easy, but just you have to think a little bit, and then you can answer it easily. So how this hydrogen bonding looks like? So this is the structure of the thymine. This is the structure of the thymine. These are the this is the phosphate linkage. These are the ribose or deoxyribose sugar. Okay, this is the sugar moiety. So this entire portion is thymine. This entire portion is guanine. This is adenine and this is cytosine. So as you can see, guanine and cytosine both are making three hydrogen bonds, and adenine and thymine, they are making two hydrogen bonds. Okay, and so that is how it looks like, the hydrogen bonds between the nucleic bases. So is it clear? I will hope that the answer is clear to you. If not, please let me know. Okay. After each and every question, if you have doubt, please let me know. Then and there, I will I will answer your query. Okay. So this was our first question. If there is no doubt, let me move to the next question. So our problem number 2 is, which of the following is known as a catalytic antibody? Your options are number A, enzymes, number B, halftone, number C, epitope, number D, paratope. Which of the following is known as catalytic antibody? I think in my previous session I have discussed in detail about epitope and paratope, and I have mentioned a little bit about the halftone and enzyme too. So in our previous session already we have discussed which of what are the things these followings are. You can check it from the last class's lecture video, which is uploaded already. So which of the following is known as a catalytic antibody? Enzyme, halftone, epitope or paratope? I will again wait for 2 minutes, then I will check the chat box. Meanwhile, let me know which of the following you think is the correct answer. Which of the following is known as a catalytic antibody? Enzyme, halftone, epitope, paratope. I will wait for 1 more minute, then I will check the chat box. See, even if your answer is wrong, I have already mentioned it does not matter, but please try to answer the questions, okay? Let me know which of the following is known as catalytic antibody: enzyme, hapten, epitope, or paratope. Okay. Let me check the chat box now. Okay, there is no answer. Okay, let me show you what is the correct answer and then let us discuss about the catalytic antibody. Okay, so the following that is known as the catalytic antibody will be, it is number A, enzymes. Okay, so enzymes, an enzyme is an antibody that expresses catalytic activity. Okay, a single molecule of an antibody, enzyme, or enzyme is capable of catalyzing the destruction of thousands of target molecules. An enzyme is used to lower the activation energy of a reaction, allowing the transition state to be possible, just similar to an enzyme, and the product to be formed. And enzymes are typically artificially made by having the immune system make antibodies that bind to a molecule that resembles the transition state of the catalytic process. Okay, so this statement is very important. Let me highlight it for you. So the enzyme is typically made and is made by and it is made by having the immune system make antibodies that bind to a molecule that resembles the transition state of the catalytic process. Okay. So what are the examples of the enzymes? The examples are as follows. The hydrolysis of the hydroxyester by enzymes. Hydroxyester forms cyclic intermediate during hydrolysis. Cyclic phosphonate ester is the structural analog of the cyclic intermediate. This analog is used as an antigen to elicit the antibodies. These antibodies bind the cyclic intermediate, increasing the reaction rate. So these are the examples of the enzymes. So one very important thing which I thought it is not directly involved in your this week's course content, but it is a one very crucial thing, okay, which is known as the antibody directed enzyme prodrug therapy. So what is a prodrug? So basically sometime what happens, a particular compound as a drug is not suitable for administration purpose or many other purposes, okay. So the what how to solve how to deal with this kind of problem? So this kind of problems can be dealt by using the prodrug therapy. So what is a prodrug? Prodrug is a compound which is needed to be activated to be the original drug, or to be the, what should I say, before the particular the drug for showing its activity, it is first activated from a particular prodrug. Okay, so that is the before the just the active drug, it is known as the prodrug. So sometimes prodrugs are given. So there is a very important thing which is known as the antibody directed enzyme prodrug therapy. So okay, so which is also abbreviated as ADAPT. Okay, so what is this? So as I have mentioned that enzymes are antibodies which have the catalytic property. Okay, it is possible that prodrugs could be designed that act as antigens for these antibodies and are activated by the enzyme's catalytic properties. This can be done by immunizing mice with the transition state analog of the reaction that is desired, followed by isolation of the monoclonal antibodies by the hybridization techniques. As the antibody targets the prodrug rather than the antigens on the cancer cell, it fails to target the drugs that kill drugs to cancer cells. Okay, fails to target drugs to cancer cells. However, it should be possible to construct the hybrid antibodies where one arm recognizes antigens on the cancer cells while the other arm recognizes the prodrug and activates it. This approach is still in the early stages of the development, but it has several potential advantages over ADEPT, which is I will discuss in some other class. Okay, for example, it should be possible to design the catalytic mechanisms that do not occur naturally, allowing the highly selective activation of the prodrugs at tumors. It also removes the risk of the immune response due to the foreign enzymes. At present, the catalytic activity of enzymes is too low to be useful, and much more research has to be carried out. Okay, so this is about the enzymes, what is the examples, and what is the antibody directed enzyme product therapy. What is epitope, what is paratope, and what is hapten? These things I have already discussed in my previous lecture. You can just go through it if you have any doubt regarding that point. So I hope it is clear. If not, please let me know. I will discuss it once more time. Otherwise, I will move to the next question if it is all right to you. Okay. Now let me go to the next question then. So the problem number 3, it says which of the following statements is true regarding the double strand DNA when heated above its melting temperature. Your options are: number A, there is a decrease in the absorption of UV light of wavelength of 260 nanometer. Number B, there is an increase in the absorption of UV light of the wavelength of 260 nanometer. Number C, there is no change in the absorption of UV light of the wavelength of 260 nanometer. Number D, it does not absorb the UV light at all. Which of the following statements is true regarding the double strand DNA when heated above its melting temperature? Number A, there is a decrease in the absorption of UV light of the wavelength of 260 nanometer. Number B, there is an increase in the absorption of UV light of the wavelength of 260 nanometer. Number C, there is no change in the absorption of the UV light of the wavelength of 260 nanometer. Number D, it does not absorb the UV light at all. Again, I will wait for 2 minutes, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer for the problem number 3. Which of the following statements is true regarding the double strand DNA when heated above its melting temperature? Which of the following statements is true regarding the double strand DNA when heated above its melting temperature? Your options are: number A, there is a decrease of the absorption of UV light of the wavelength of 260 nanometer. Number B, there is an increase in the absorption of UV light of the wavelength of 260 nanometer. Number C, there is no change in the absorption of UV light of the wavelength of 260 nanometer. Number D, it does not absorb the UV light at all. Which of the following statements is true? Okay, let me check the chat box. Let me see if there are any answers for the problem number 3. Okay, so there is no answer for problem number 3 also. Okay, let me show you and let us discuss. Please try to answer during the class. Okay, even if your answer is wrong, I am saying it repeatedly that it does not matter, but please try to answer the question if it is possible. Or you can, because even if your answer is wrong, it does not matter. Okay, so the statement that is true regarding the double strand DNA when heated above its melting temperature will be, it is number B. There is an increase in the absorption of UV light of the wavelength of 260 nanometer. So what is the melting temperature of DNA? This is very important, that what is the melting temperature of the DNA? Okay, the temperature at which half of the DNA strands are single stranded and half are the double stranded. The melting temperature characterizes of the DNA composition. Higher the GC content and higher the melting temperature due to the higher more number of hydrogen bonds. So this is very important thing that you have to remember, that higher the guanine and cytosine content in a particular DNA sample, its melting temperature will be higher. Why? Because there are more hydrogen bonds, and more hydrogen bonds means it will take more temperature or more energy to break those hydrogen bonds to make the double strand DNA to the single strand, right? What is the calculation? What is the formula? So one formula is goes like this: base composition is is to melting temperature equal to 59.9 plus 0.41 into percentage of guanine cytosine minus 600 divided by bond length. So this is a calculation, but there are more other accurate methods are also available. You don't need to remember what is the particular calculation or particular formula is, but you have to just know what is the melting temperature, what is the significance. So the graph looks like this. Okay, on the y-axis we have the absorption of 260 nanomolar light. We have the absorption like from 0 point, and here we have the temperature. So as you can see, the melting temperature of a DNA, it is in this position. Okay, it's around you can see that for the double stranded DNA, it is around the 82.5 or to 83. Okay, so as you can see, as there is a sharp increase in the absorption of UV light of the wavelength 260 when the DNA double stranded DNA is heated above its melting temperature. So as you can see, as we are increasing the temperature from 75 to 80, there is very a slow increase of the absorption. Okay, on the line, this is the up to this it is flat, and there is a little increase of the absorption, but as it is crossing the melting temperature, there is a sharp increase in the absorption, as you can see for the double stranded. Okay, so that indicates that this thing, that there is an increase of the absorption of UV light of the wavelength of 260 nanometer once the double strand DNA is heated above this melting temperature. Okay. So I hope it is clear. If there is any doubt, you can let me know, right? Otherwise, let me go to the next problem if there is no doubt in problem number 3. Okay. So now let me go to the next problem. Problem number 4, it says that the structure of DNA is closely related to its biological function. Which of the following statements regarding the structure of DNA is correct? Your options are: number A, all base pairs interact by forming 3 hydrogen bonds. Number B, DNA is composed of nucleotide subunits, each containing a base, a deoxyribose sugar, and a phosphate group. Number C, the double helix of DNA is stabilized by the interactions between the adjacent ribose sugars. And number D, each DNA sequence folds into a unique and complex 3D structure. This will be structure. Okay, there is a spelling mistake. I will correct it later. Read the options very carefully and then let me know which of the following statements regarding the structure of DNA is correct. Whenever there is a question of this correct, incorrect, true, false type of thing, please be very very clear about the options. We read all the options very carefully, because I have seen most of the students they making mistakes in this type of correct, incorrect, true, false questions. The structure of DNA is closely related to its biological function. Which of the following statements regarding the structure of DNA is correct? Number A, all base pairs interact by forming 3 hydrogen bonds. Number B, DNA is comprised of the nucleotide subunits, each containing a base, a deoxyribose sugar, and a phosphate group. Number C, the double helix of DNA is stabilized by interactions between the adjacent ribose sugars. Number D, each DNA sequence folds into unique and complex 3D structures. I will wait for 2 minutes, then I will check the chat box. Please let me know A, B, C, or D, which of the following you think is the correct answer. The structure of DNA is closely related to its biological function. Which of the following statements regarding the structure of DNA is correct? I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which of the following you think is the correct answer. Okay, let me check the chat box. So I got one answer which is saying it is number B. Let me check if number B is the correct answer or not. So the correct answer will be yes, it is number B, which is mentioned that DNA is comprised of the nucleotide subunits, each containing a base, a deoxyribose sugar, and a phosphate group. So the first thing, let us know what is the, let us understand what are the nucleic bases of the DNA. I have already, I think, mentioned in my first slide that there are four different nucleic acid bases for the DNA: adenine, guanine, thymine, and cytosine. In case of the RNA, there is no thymine, there is only the particular uracil. Okay, I will let you know what is the structural difference between these two. So these adenine and guanine, these are known as the purines. They are the bicyclic, they are bicyclic rings. Okay, the one basically you can remember like this. In case of adenine and guanine, these are the purines where one pyrimidine ring is clubbed with a imidazole ring system, right? And the numbering starts from here and goes like this. And in case of guanine, the structure is it also has a purine ring, but the other functional groups are little different. This is the structure of cytosine. Okay, and these are the pyrimidine bases, the cytosine and thymine. In case of uracil, we don't have this methyl group. Okay, in case of uracil, there is no, this methyl group is not there. Otherwise, the structures are same. So adenine, guanine, cytosine, thymine, these are the nucleic bases, acid bases of DNA. Adenine makes hydrogen bonds with thymine, guanine makes hydrogen bonds with cytosine. This I have already mentioned. Now there is one thing which is called the nucleosides. So what is the nucleosides? So nucleosides are the building blocks of the DNA. So nucleosides are the these are the are basically the nucleic bases, nucleic acid bases, and which is attached with the deoxyribose sugar. So this is known as the deoxyribose sugar. Ribose because it has this particular five-member ring. Okay, and it is known as 2-deoxyribose because at the two position, 2 position, we don't have any oxygen. Okay, that is why it is known as the deoxyribose, the deoxy thing. Okay, deoxyribose base. Deoxy sugar. So in case of RNA, this is not the deoxy sugar is not there. There is only ribose sugar is present. Okay. So this is this particular ribose particular this deoxyribose sugar and the nucleic base, they two go together. They are known as the nucleoside. So this is the D-deoxy adenosine, D-deoxy guanosine, D-deoxy thymidine, and D-deoxycytosine. So these are the nucleic side. Now we have the nucleotides. What are the nucleotides? Nucleotides are basically the this particular base, the deoxyribose sugar and phosphate groups together. These are known as the nucleotides. So that's how this particular this thing happens. Like this is for example, this is the there is a phosphate linkage between two ribose sugar, and this totally it is called the nucleotide. Okay. So this is the linkage through the nucleosides to the phosphate group. Now, this is known as the secondary structure of DNA. If you remember, we have when we were discussing about the amino acid and peptide linkage and these things, we have learned about the primary structure of the particular primary structure of the protein, secondary, tertiary, and quaternary structure. So this is the almost similar thing like with the DNA or nucleic acids also. We have a secondary structure of DNA which is known as a double helix. Okay, DNA double helix, and it is much looks like the alpha helix of our amino acids, right? You can see there are 3 hydrogen bond between G and C and 2 hydrogen bond between A and T. The distance between between two different this strands is 10 Armstrong, and for the similar strand the distance is 34 Armstrong. This is known as the distance between two minor group. Actually, the distance between two minor group is 10 Armstrong, and between two major group is 34 Armstrong. Okay, this from this to this is known as the major group, and this to this is known as the minor group of DNA. So these are the things regarding DNA, and the option number B was the correct. So I think it is fine, or is there any doubt? If there is any doubt, please let me know. Okay. So since there is no doubt, I will move toward the next problem. Problem number 5, it says that which of the following enzyme is crucial for the tertiary structure of the DNA. Your options are number A topoisomerase, number B helicase, number C DNA polymerase, and number D ligase. Which of the following enzymes is crucial for the tertiary structure of the DNA? Topoisomerase, helicase, DNA polymerase, or ligase? Let me know in the chat box A, B, C, or D which of the following is the correct answer. Which of the following is the enzyme that is crucial for the tertiary structure of DNA? Like the proteins and the amino acids, the peptide linkage, the DNA also have a tertiary structure, and there is a very important, very crucial enzyme that is responsible for keeping the tertiary structure of DNA stable. What is that particular enzyme? Topoisomerase, helicase, DNA polymerase, or ligase? Let me know A, B, C, or D which of the following you think is the correct answer for problem number 5. Which of the following enzymes is crucial for the tertiary structure of DNA? Which of the following enzymes is crucial for tertiary structure of DNA? Topoisomerase, helicase, DNA polymerase, or ligase? Let me know A, B, C, or D which of the following you think is the correct answer. Let me check the chat box. Let me check if there is any answer for this problem number 5. Okay, there is no answer till now. Okay, let me show you what is the correct answer and then let us discuss about it. Okay, so the enzyme that is crucial for the tertiary structure of DNA will be, it is topoisomerase, number A. Let us now discuss what is the topoisomerase enzyme. So just like I have mentioned earlier, we have discussed this is the secondary structure of DNA, right? So the DNA secondary structure is almost similar to the secondary structure of our protein, the alpha helix. You can just have a similarities between these two, the double helix of DNA and alpha helix of the protein. So like the proteins, the DNAs also have the tertiary structure. Okay. So, the tertiary structure of DNA is often neglected or ignored, but it is important to the action of the quinolone group of antibacterial drugs and to several anti-cancer agents also. These things, this quinolone group of antibacterial and anti-cancer agents, we will discuss in our later into our course. We will discuss it. You can remember this name of this particular enzyme because we will have lots of discussion around it. DNA is an extremely long molecule, so long in fact that it would not fit into the nucleus in the cell if it is existed in a linear molecule. Okay, it has to be coiled up into more compact three-dimensional shape which can fit into the nucleus, a process known as the supercoiling. So this is very important. The supercoiling, what is the need of supercoiling? That to keep the DNA, for making the DNA fit into the nucleus, we need this supercoiling, right? This process requires the action of a famous enzymes known as the topoisomerases, which can catalyze the seemingly impossible act of passing one stretch of DNA helix to another. Now, they do it by the temporarily cleaving one or both strands of the DNA helix to create a temporary gap and then resealing the strands once the crossover has taken place. Okay, the supercoiling allows the efficient storage of DNA, and the DNA has to be uncoiled again for replication and transcription, and this process to take place. If the uncoiling did not take place, the unwinding process catalyzed by the helicase enzyme that takes place during the replication, the replication transcription would lead to the increased transtension due to the increased supercoiling of the remaining DNA double strand. You can demonstrate this principle by pulling apart the strands of rope or the sisal. The same topoisomerase enzyme is responsible for catalyzing the uncoiling process. So inhibition of this enzyme would effectively block the transcription and replication. Topoisomerase 2. So there are two types of topoisomerase, mainly, majorly. Okay, topoisomerase. Okay, just to give. Yeah. There are mainly two types of topoisomerase. Topoisomerase 2 is a mammalian enzyme that is crucial for the effective replication of DNA. The enzyme binds to the parts of DNA where two regions of the double helix into the near proximity. The enzyme binds one of these DNA double helixes, and tyrosine residues are used to nick the DNA strands, and this results in temporary covalent bond between the enzyme, resulting 5' prime to each strand. The stabilizing the DNA, the strands are now pulled opposite direction to form a gap through which the intact DNA region can be passed. This enzyme then reseals the strands and departs. Topoisomerase 1 is similar to topoisomerase 2 in that it relieves the torsional stress in the supercoiled DNA during the replication, transcription, and repair of the DNA. The difference is it cleaves only one strand, whereas the topoisomerase 2 cleaves the both strands. The enzyme catalyzes a reversible transesterification reaction similar where the tyrosine residues enzyme is linked to 3' phosphate and end of the DNA rather than 5' end. This creates a cleavable complex with a single strand break. Relaxation of the torsional strain takes place by allowing the intact strand of the pass through the nick between the free rotation of the DNA. Now, once the torsional strain is relieved, the enzymes rejoin and reseals. Topoisomerase 4 is also there, which is a bacterial enzyme that carries out the same process as the mammalian enzyme topoisomerase 2, and it is an important target for the fluoroquinolone antibacterial drugs. So this is a visual representation how this topoisomerase 2 catalyzes the crossover of the DNA strands. Okay. So this is the DNA. There are two different double helix of DNA as you can see, and both are in the supercoiled position. So what this topoisomerase 2 will do, it will cleave the particular DNA's double helix in the two in the both strands. As you can see, it is cleaving in this strand also, this and that strand also. And by cleaving in both strands, it creates a gap through which the intact strand it can pass crossover, and after that it religases and it relaxes the cleaved strands and it leaves. Okay, that is how it relaxes. And there is a tyrosine residue in this topoisomerase 2 enzyme, which actually attacks on this phosphate linkage for creating the gap, and this is the mechanism by which the topoisomerase 2 splits the DNA chain. Okay. So this is about how the topoisomerase actually relaxes the supercoil, and which is very favorable for the tertiary structure of the DNA. Okay. So is it clear or is there any doubt? If not, I will move to the next problem. So if there is no doubt, I will move toward the next problem. Please let me know if there is any problem through the chat box. Okay. I'm monitoring the chat box, and also please try to answer the questions. Okay. Even if your answer is wrong, it does not matter, but try to answer it. Okay. Now let us go to the next problem. Problem number 6, it says that the combination of the chemical reactions for only C-specific cleavage are, your options are number A, hydrazine and piperidine, number B, DMS, dimethyl sulfide and piperidine, number C, DMS and dilute acid, you can say H plus, and piperidine, number D, hydrogen, aqueous, NaCl and piperidine. Let me tell you that this question is related to the Maxam-Gilbert method of the DNA sequencing. Okay. Particular combinations of chemical reactions are used to cleave specific bases. You have to identify which of the particular following combination is specific for the cleavage of the cytosine. Your options are number A, hydrazine piperidine, number B, DMS piperidine, number C, DMS dilute HC and dilute acid and piperidine, number D, hydrogen aqueous NaCl and piperidine. Let me know A, B, C or D, which of the following is the correct answer. I will wait for 1 to 2 minutes, then I will check the chat box. After checking the chat box, I will discuss this particular question in detail. But before that, please try to answer the question. Even if your answer is wrong, I have mentioned it does not matter. But please try. The combination for the chemical reactions for only species specific cleavage are number A, hydrogen and piperidine, number B, DMS and piperidine, number C, DMS, dilute proton, dilute acid and piperidine, and number D, hydrogen, aqueous NaCl and piperidine. Here C indicates, or C means the cytosine. Okay. A, B, C or D, which of the following is the correct answer? Okay, let me check the chat box. Let me check if there are any answers. Okay, I got one answer, which is saying that it is number D. Let me check if number D is the correct answer or not. Okay, so I got one answer. Let me check if it is correct or not. Number D. So the correct answer will be the combination of the chemical reactions for only cytosine specific cleavage are very good. It is number D, hydrogen, aqueous NaCl and piperidine. So basically, this is the Maxam-Gilbert sequencing methods of DNA. So what is the principle? The core idea behind the Maxam-Gilbert sequencing is to create a set of the DNA fragments of varying lengths, each ending at a particular type of the base, adenine, thymine, cytosine and guanine, by using base-specific chemical reactions. These fragments are then separated by size using gel electrophoresis, and then the DNA sequence can be deduced by analyzing a pattern of the separated fragments. So what are the key steps? The key steps are the DNA preparation. The DNA fragment to be sequenced is purified, denatured into the single strands. One end of the fragment is then radioactively labeled, typically with the 32 phosphorus. Then chemical cleavage reactions. The labeled DNA is then divided into 4 reaction tubes, each with a specific chemical that modify and cleave the DNA at different bases. For the guanine cleavage, it is achieved by methylating the guanate by dimethyl sulfide, followed by the hot piperidine treatment that cleaves the DNA backbone. For cytosine, using the hydrogen in presence of 1.5 molar NaCl and subsequent piperidine treatment. Cytosine and thymine together, the cleavage is achieved during the hydrogen piperidine without NaCl. Okay, and guanine and adenine cleavage used by formic acid along with the piperidine. So this concentration of modifying chemicals is carefully controlled to ensure the average only one modification occurs per DNA molecule. Then the third step is the gel electrophoresis. The resulting fragment of the 4 reaction are then separated side by side by on the denaturing polyacrylamide based on their size. The smallest fragment migrate fastest are found at the bottom of the gel. Autoradiography and sequence preparation. The gel is then exposed to the X-ray film to visualize the radiolabeled DNA fragments. The sequence is read from bottom to top, inferring the base at the each position, observing the presence and absence of the band for different lengths. So I will show you how to do this Maxam Gilbert kind of sequencing. I will show you in today's class itself. Just for now, just remember these are the specific cleavage that are achieved by the by a specific chemical reactions. Okay. So the correct answer was yes, it was number D. For the cytosine specific cleavage, we have to use hydrogen in presence of 1.5 molar NaCl and with piperidine treatment. Okay. Now let me go to the next problem. Problem number 7, it says that the term hyperchromic shift indicates your options are as follows. Number A, decrease in the absorption of the UV light by a solution of DNA. Number B, increase in the absorption of UV light by a solution of DNA. Number C, increase in the shift of the wavelength of UV light by a solution of DNA. Number D, decrease in the shift of the wavelength of the UV light by the solution of DNA. Again, I will repeat the term hyperchromic shift indicates number A, decrease in the absorption of UV light by a solution of DNA. Number B, increase in the absorption of UV light by a solution of DNA. Number C, increase in the shift of the wavelength of UV light by a solution of DNA. And number D, decreased in the shift of the wavelength of UV light by a solution of DNA. Which of the term hyperchromic shift indicates which of the following? Let me know A, B, C or D which of the following is the correct definition for the hyperchromic shift. Again, I will repeat the question. The term hyperchromic shift indicates the decrease in the absorption of UV light by a solution of DNA. Number B, increase in the absorption of UV light by a solution of DNA. Number C, increase in the shift of the wavelength of UV light by a solution of DNA. And number D, decrease in the shift of the wavelength of UV light by a solution of DNA. I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following is the correct answer. The term hyperchromic shift indicates which of the following? Let me check the chat box. Okay, I got one answer which is saying it is number B. Okay, I got two answers. Both are saying it is number B. Let me check if number B is the correct answer or not. So, the term hyperchromic shift, it indicates, very good, it is number B, increase in the absorption of UV light by a solution of DNA. So this graph basically it indicates how this particular it is on the y-axis it is the absorption, on the x-axis it is the wavelength. This is the absorption maxima, as you can see like this. Okay, so by the increasing of the increasing in the absorption, okay, it is known as the hyperchromic shift. Means when the particular, if the graph was particular like this, it is known as the hyperchromic shift, which is increasing increasing the particular absorbance, right? If it looks like this, it known as the hypochromic shift, which is decrease of the absorption. Okay, and what is the there are two more things which you have to remember. So one is the bathochromic shift and one is the hypsochromic shift. Bathochromic shift or the red shift, when the maxima shifts to the higher wavelength like this, it is known as the absorption maxima shifts to the higher wavelength, it is known as the bathochromic shift or red shift. And if the absorption maxima it shifts to the lower wavelength, it is known as the hypsochromic shift or the blue shift. Okay, so these are the terms which you need to remember, which you need to understand. Okay, so the hyperchromic shift is due to the shift is due to the increase of the intensity or absorption Emax to increase due to the introduction of oxochrome for like for example piperidine. From piperidine it is 257 nanomolar and the particular Emax is 2750, but 2-methyl piperidine it is 262 nanomolar and the Emax is 3560. Hypochromic shift, inverse of the hyperchromic shift, that is the decrease of the intensity. Introduction of any group to the compound which is going to alter the molecular pattern of the compound results in the hypochromic shift. For example, biphenyl absorbs at 250 nanomolar and 19,000 is the Emax. Whereas 2-methyl biphenyl has an absorption of 237 nanomolar and the absorption maximum is 10,000 to 250. So these are the terms which you need to remember: the hyperchromic, hypochromic, bathochromic and hypsochromic. Okay. So is it clear or is there any doubt for this problem number 7? Problem number 7 is I think there is no doubt and you have answered it correctly. So very good. Now let me go to the next problem if there is no doubt in this question. Problem number 8 says, which of the following statement is false regarding the DNA? Your options are: number A, all the A DNA, B DNA and Z DNA have right-handed helix. Number B, A DNA is more compact than B DNA. Number C, most common DNA is B DNA. And number D, A DNA has 11 base pairs in a complete turn. Which of the following statement is false regarding DNA? So DNAs are also, there are classifications. Mainly there is a DNA, B DNA and Z DNA. And this different forms of DNA has some crucial characterize or crucial and particular characteristics. Now you have to identify which of the following statement is false regarding the DNA. Your options are: number A, all the A DNA, B DNA, Z DNA have the right-handed helix. Number B, A DNA is more compact than B DNA. Number C, most common DNA is the B DNA. And number D, A DNA has 11 base pairs in a complete turn. Let me know in the chat box A, B, C or D which of the following you think is the false statement regarding the DNA. I will wait for a minute or two. Then I will check the chat box. Read all the options very carefully because I have seen most of the students, they very easily make mistake in this kind of true false and of correct incorrect question answers. Which of the following statements is false regarding DNA? Which of the following statements is false regarding the DNA? Let me check the chat box. Okay, there is no answer. Okay, it's fine. Let me show you what is the correct answer and let us discuss about this different forms of the DNA. So the statement which is false regarding the different forms of DNA will be: number A, all the ADNA, BDNA and ZDNA have the right-handed helix. This statement is false. Now let us see what are the different structural forms. So this is known as the ADNA, which is the most I think common one. Okay, then we have this the most common one is the BDNA. We have ADNA, which is little compact than the BDNA, as you can see here, and then we have this ZDNA, which is little bit deformed. So this is the topmost view. This is the ADNA, this is the BDNA, which is the most common form. This is the ADNA, which is little compact, and this is the ZDNA, which is little it is more expanded. You can see. Okay. So what are the characteristics? First is the most important is the helical sense. Okay. The A form of the DNA is the right-handed helix. The B form is also right-handed helix, but the Z form is left-handed helix. The diameter. The diameter of the A form is 26 Armstrong. The for B form it is 20 Armstrong. From the Z form it is 18 Armstrong. The rise per turn of the helix, it is the 28 Armstrong for ADNA, 36 Armstrong for BDNA and 44 for ZDNA. This is basically I have shown you in the our so this is the this rise per turn of the helix. It means the distance between the this is the distance between the two major groups. Okay, so it is for 26 Armstrong in case of ADNA, 36 for the BDNA, which is the most common one, and for ZDNA it is 44 Armstrong. Base pairs per helical turn is 11 base pairs in case of the ADNA, 10 base pairs in case of B, and 12 base pairs in case of the ZDNA. Helix rise per base pair is 26 Armstrong, 2.6 Armstrong, and it is for 3.6 Armstrong in case of B and 3.7 in case of the Z. Base tilt normal to the helix is 20 degree as from the Z A form. For the B form it is 6 degree. For the Z form it is the 7 degree. Glycyl bond conformation is anti in case of the ADNA, anti in case of the BDNA also, and it is anti in case of the pyrimidine bases and syn for the purine bases in case of the Z form of the DNA. So these are different characteristics and different differences between the forms of the DNA A, B and Z DNA. But remember, BDNA is the most important or most crucial form of the DNA. This is very important which you need to remember. Okay. Now I will go to the next problem. So problem number 9, it says the restriction enzyme BAMHI cleaves the base pairs of number A and T, number B, G and G, number C, A and C, and number D, A and A. Restriction enzyme BAMHI cleaves between the base pairs of A and T, G and G, A and C, and A and A. Where this particular restriction enzyme cuts a particular DNA strand. The restriction enzyme BAMHI cleaves between the base pairs of Which of the following? Number A, between A and adenine and a thymine. Number B, between two guanine bases. Number C, between adenine and cytosine. And number D, between two, it cuts between two adenine bases. Which of the following will be the correct answer? The restriction enzyme MAMI lives between which base pairs? Let me check the chat box. Is there any answer? Okay. There is no answer, but let me show you what is the correct answer and let us discuss about it. So the restriction enzyme BAMHI, it cuts between the base pairs of number B, G and G. So what is the restriction enzyme? Today I have discussed a little bit about the restriction enzyme, but I will discuss it in detail in our upcoming class. Okay. So basically remember that the restriction enzymes are the enzymes that cuts the DNA at specific nucleotide sequences known as the restriction sites. It is found in bacteria, have evolved to provide the defense mechanism against the invading viruses. In bacteria, they selectively cut off foreign DNA in a process called restriction. To cut the DNA, restriction enzyme makes two insertions, each strand of DNA double helix. So the restriction enzymes, restriction endonucleases digest the double-strand DNA at specific sites. Recognition site is the palindrome. Palindrome means it will be same from by reading like from forward or backward. Okay, and for example, madam, I am madam. This line, if you read it from this side and this side, it will be the same. These are known as the palindrome sequence, and these palindrome sequences are the recognition side of the restriction enzymes. It reads at the same on the both 5 prime to 3 prime. Like for example, you can see from 5 prime to 3 prime, it looks like 5 prime AGCT 3 prime, and its complementary strand is also like from 5 prime, it is AGCT and 3 prime. Okay, so these are known as the palindrome sequences. Recognition sites are different for sizes and unique sites. They cuts between the same nucleotides on the each strand. Okay, and it is so for example, these are the examples of the restriction enzyme, and you can see BAMI, it cuts between the G and G. One more thing, that this ALUI it and HAE triple I, it produce the blunt ends and others are producing the sticky end. So what are the blunt ends, what are the sticky ends, all these things I will discuss in our next class. Okay, this is not entirely false in our today's course content, but I thought to give you an overview of it little bit. We will discuss it in detail in our next class. What is the restriction enzyme? There are many restriction enzymes which are very crucial. These things I will discuss in detail in the next class. Okay, for now you just remember that what is the restriction enzyme and these top points. Okay. Now let me go to the next problem. Problem number 10 says, which of the following is an implication of high GC base pair concentration in the DNA? Your options are number A, high thermal stability, number B, lower gene expression level, number C, lower gene density, number D, less challenge in the PCR and DNA sequencing. Which of the following is an implication of high GC base pair concentration in DNA? Option number A, high thermal stability, number B, lower gene expression level, number C, lower gene density, and number D, less challenge in the PCR and DNA sequencing. Which of the following is an implication of high GC base pair concentration in DNA? I have already discussed a little bit about it. If you remember, in our previous one question, I have discussed a little bit about it. If you remember, then you can easily answer this question. Try whatever you think is the correct answer. Let me know in the chat box A, B, C, or D, which of the following you think is the correct one. Even if your answer is wrong, it does not matter, but please try to answer. Let me know which of the following is an implication of high GC base pair concentration in DNA. High thermal stability, low gene expression level, low gene density, or less challenge in PCR and DNA sequencing. I will wait for a minute, then I will check the chat box and I will show you the answer. Which of the following is an implication of high GC base pair concentration in DNA? Let me check the chat box now. Okay, so I got one answer which is saying it is number D. Let me check if number D is the correct answer or not. So, the implication of the high GC base pair concentration in DNA, the correct implication will be it is number A, high thermal stability. If you remember that in our previous question when we were discussing about the DNA melting temperature, I don't know which one was that. I think this one. So I have mentioned that the higher GC concentration will have the higher melting temperature due to the more hydrogen bond. Okay, so this is related the same, the question, both questions are related in the same way. Okay, so the higher the GC base pair concentration, higher thermal stability will be there, higher will be the melting temperature of our DNA. Okay. So, what are the implications of the high GC concentration? That's the thermodynamic stability. GC base pairs from three hydrogen bond compared to the two between adenine and thymine, resulting in higher stability and higher melting temperature required to denature the DNA duplex. Impact on gene expression and regulation. GC-rich promoters may be more active and lead to the higher gene expression levels. High GC content can also influence the binding of the transcription factors affecting the gene regulation. In fact, the studies have shown that high guanine and cytosine content increasing mRNA levels in the mammalian cells. Genomic organization and features. GC-rich regions are often associated with the higher gene density and compactness, earlier replication timing, the higher combination rates compared to the GC pure regions. They can also be indicative of the gene's origins from a foreign genome, potentially through the horizontal gene transfer. Genome mapping and disease development. GC-rich regions are crucial for the gene mapping due to their high gene density. Analyzing CpG island rich GC regions can also help to study and characterize genetic disorder. For example, Huntington disease is linked to the changes of the number of CAG repeats in the HTT gene, which has GC rich repetitive areas called the CpG islands. Challenges in molecular biology technique. High GC content in the DNA templates can present challenges processes like PCR and DNA sequencing. This is because high GC content requires higher denaturation and annealing temperature during PCR, which can increase the chances of non-specific binding and false positive. It can also create a stable secondary structure that interfere with the DNA polymerase activity. For example, in PCR, primers with the high GC content greater than 60% necessitate a higher annealing temperature, which can make it challenging to optimize the reaction for optimal specificity and efficiency. Okay, so these are the implications or these are the things that will happen if your DNA sample it contain high GC concentration. Okay, the GC concentration mainly impacts on the thermodynamic stability, gene expression level, genomic organization and features, genome mapping, disease development, and also in the molecular biology technical techniques like PCR. I will go to the next problem. Problem number 11. Which of the following statements regarding albinism is false? Your options are number A, it is associated with a genetic illness. Number B, it results in non-expression of a particular proteins and expression of defective proteins. Number C, it is associated with a deficiency of an enzyme known as tyrosinase. Number D, tyrosinase is an iron-containing enzyme that catalyzes the synthesis of melanin. Which of the following statements regarding albinism is false? Your options are number A, it is associated with a genetic illness. Number B, it results in a non expression of a particular protein or expression of the defective proteins. Number C, it is associated with a deficiency of an enzyme called tyrosinase. Number D, tyrosinase is an iron containing enzyme that catalyzes the synthesis of melanin. Which of the following statements regarding albinism is false? Let me know in the chat box A, B, C or D which of the following you think is the correct answer, means which of the means the false statement regarding albinism. I will wait for a minute or two, then I will check the chat box. I will repeat the options. Number A, it is associated with a genetic illness. Number B, it results in non-expression of a particular proteins or expression of a defective proteins. Number C, it is associated with a deficiency of an enzyme called tyrosinase. And number D, tyrosinase is an iron-containing enzyme that catalyzes the synthesis of melanin. Which of the following statements regarding albinism is false? Let me check the chat box. Let me check if there is any answer. Okay, I got one answer which is saying it is number D. Let me check if number D is the correct answer or not. Okay, so let us see what will be the correct answer. So the statement which is false regarding the albinism will be very good. It is number D, which says that tyrosinase is an iron containing enzyme, the enzyme that catalyzes the synthesis of melanin. Okay, there is a typographical error. Correct it. Yeah. So the correct answer is number D. Let me just discuss once. Okay. So this is the structure of the tyrosinase. Okay, if you can see here, you will find there are two copper centers. Okay, so tyrosinase is basically a copper containing enzyme that catalyzes the synthesis of melanin. It has there are two copper A and copper B. Copper A is bound to the two histidine residues, and one phenylhalidine residues is also there, and there is for copper B it is bound with a histidine three histidine residues, one histidine 259, histidine 263 and histidine 296, and there is also phenylalanine at the vicinity of this enzyme, and both enzymes are bridged with a OH, the hydroxyl group. So albinism is a genetic condition characterized by the partial or complete absence of the pigment melanin, as you can see like this, okay, in skin, hair and eyes. The lack of the pigment can lead a range of vision problems and increased sensitivity to sunlight. Now, it is a lifelong condition, and with a proper management, individual with albinism can live full and healthy lives. Key characteristic: reduced and absence of melanin. Melanin is a pigment that gives the color to the skin, hair and eyes. People with albinism have a little very little or no melanin. Oculocutaneous albinism, it is the most common type affecting the skin, hair and eyes. Ocular albinism, its type primarily affect the eyes, with the skin and hair appearing similar to family number. Vision problem: albinism is often associated with the various vision issues, including the nystagmus, strabismus, photophobia or reduced visual acuity. Okay, and then increased risks of the skin cancer due to the lack of melanin's protective effect against the UV radiation. Causes: genetic mutation. Albinism is caused by mutation in the genes involved in melanin production. Autosomal recessive inheritance. In most of the cases, albinism is inherited in an autosomal recessive manner, meaning a person needs to inherit two copies of the mutated gene, one from each parent, to have the condition. Management or treatment: there is no cure of the albinism. Eye care, regular eye exams, glasses or vision aids can help to manage the vision problem. The screen protection: sunscreen, hats, and protective clothes are essential to prevent the sunburn and skin cancer. Genetic counseling helps individuals and families to understand the condition and inheritance pattern. Okay, so these are the key characteristic causes of the albinism. So yeah, correct answer was number D, which says it is an iron containing enzyme, but it is not, it is a copper containing enzyme. Then I will go to the next problem. Problem number 12, which says that which of the following enzymes is used to convert the ATP to AMP in pyrosequencing? Your options are number A, isomerase; number B, polymerase; number C, sulfhydrylase; number D, apirase. Which of the following enzymes is used to convert the ATP to AMP in pyrosequencing? Isomerase, polymerase, sulfhydrylase or apirase? I will wait for a minute or two, then I will check the chat box. After that, I will discuss what is pyrosequencing in detail. Okay. Till then, let me know what is the correct answer. Let me know in the chat box, A, B, C or D, which of the following enzyme is used to convert ATP to AMP in pyrosequencing? Isomerase, polymerase, sulfurylase or apirase? Which of the following enzymes is used to convert ATP into AMP in pyrosequencing? I will wait for a minute, then I will check the chat box. Which of the following enzymes is used? Let me check the chat box because I don't, we have lots more question to discuss. Okay, let me check if there is any answer. Okay, I got one answer. It is saying number B. Let me check if number B is the correct answer or not. So the enzyme that is used to convert ATP to AMP during pyrosequencing will be, it is actually number D, apirase. Okay. So what is pyrosequencing? So pyrosequencing is a method of the DNA sequencing that utilizes a sequencing by synthesis approach combined with the luminometric light detection to determine the sequence of the nucleotide in the DNA strand. Unlike Sanger sequencing, which relies on the chain termination, pyrosequencing relies on the detection of the pyrophosphate release and the subsequent light generation when the nucleotides are incorporated into the growing DNA strand. Okay. Now let us see what is the pyrosequencing and how it is done. So there are four different steps for the pyrosequencing. The step one, the first step is the amplification of the DNA strand. Okay, first is the DNA strand is amplified and one strand is biotinylated. Biotinylated means incorporation of the biotin to the particular strand. Okay, biotinylated strand is hybridized to the sequencing primer, and primer plus templates is incubated with DNA polymerase. So one important enzyme is DNA polymerase. DNA polymerase is there, but it does not actually converts that particular ATP to AMP. Okay, so there are different enzymes which are used in case of the pyrosequencing. These are the DNA polymerase, ATP sulfurylase, luciferase, and we have apirase. So these four different enzymes, these are used in case of the pyrosequencing. Now first this the primer plus template is included with these four enzymes, then DNTPs are added, If complementary to the base, DNA polymerase will add the DNTP to the sequencing primer. For each DNTP incorporated, equimolar pyrophosphate will be added. Okay, then this pyrophosphate will be converted to ATP, and in presence of ATP, luciferin will be converted to oxyluciferin. This is also important. In presence of ATP, luciferin will be converted to the oxyluciferin, and the reaction generates light. So as you can see, APS plus PPI, it gives us the ATP with the help of the sulfurylase. Okay, sulfurylase will catalyze the conversion of the pyrophosphate to ATP, and then ATP will generate light with the help of the luciferase, which will convert luciferin to oxyluciferin. So these two steps will happen together: the conversion of luciferin to oxyluciferin and the conversion to ATP to light by the enzyme luciferase. Okay. So these are the enzymes which are the important: polymerase, sulfurylase, luciferase. Then light is detected using a sensor like this, and the peak is created. The height of the peak is proportional to the amount of the DNTP incorporated. Apide comes there, and it degrades the NTP to degrades the unincorporated DNTP and ATP. So as you can see, apide is here. Apide converts the DNTP, which is unincorporated, to DNDP, DNMP, and phosphate, and it converts ATP to ADP, AMP, and phosphate. Okay, so that is the role of the apide. And then step 4 is the process continues. The complementary DNA sequence is elongated, and the nucleotide sequence is determined from the peaks by a pyrogram. So this is a pyrogram from where we have this sequence, which will help us to determine what is the particular sequence. So remember, pyrosequencing is the method where there are 4 different enzymes are involved. We have this DNA polymerase, which incorporates the particular DNTP to the growing sequence if it finds the complementary base. Then we have sulfurylase, which converts the APS and PPI to ATP. Now we have this luciferase, which converts the ATP to light and also luciferin to oxyluciferin. We have the apidase, which break down the unincorporated DNTP and ATP, DNTP to DNDP, DNMP and phosphate, and ATP to ADP, AMP and phosphate. And this is the last step. So these are the different steps of the pyrosequencing. I hope it is clear. If not, you can let me know. I will repeat these things. Okay. So now I will go to the next problem. If it is clear or if there is no doubt regarding the pyrosequencing, then I will go to the next problem. So the problem number 13, it says that which of the following statements regarding the genetic polymorphism is false? Your options are: number A, personalized medicines are required to combat this disease. Number B, it occurs due to the mutation of the gene. Number C, the genome mapping of the patient is not required to kill the disease. And number D, this disease leads to the different individual having subtly different gene sequence. Which of the following statements regarding the genetic polymorphism is false? Number A, personalized medicines is required to combat this disease. Number B, it occurs due to the mutation of the gene. Number C, the genome mapping of the patient is not required to cure the disease. Number D, the disease leads to the different individuals having subtly a different gene sequence. Which of the following statements regarding the genetic polymorphism is false? Number A, personalized medicines are required to combat this disease. Number B, it occurs due to the mutation of the gene. Number C, genome mapping of the patient is not required to cure the disease. And number D, the disease leads to the different individuals having subtly different gene sequence. Let me know in the chat box A, B, C or D which of the following statement is false. Which of the following statements regarding genetic polymorphism is false? Number A, personalized medicines are required to combat the disease. Number B, it occurs due to the mutation of the gene. Number C, the genome mapping of the patient is not required to cure the disease. And number D, the disease leads to different individuals having subtly different gene sequence. Let me check the chat box. Okay, I got one answer which is saying it is number A. Now let me check if number A is the correct answer or not. So the statement which is false regarding genetic polymorphism will be, it is number C. The genome mapping of the patient is not required to cure the disease. So genetic polymorphism is naturally occurring variants in the gene structure that occur in more than 1% of population. Polymorphisms may influence a drug's action by changing its pharmacokinetics or its pharmacodynamics. It is also known as the intersubject variability. It paves the path for the individualized rational dosage regimen. Differences is observed in the metabolism of the drug among the different races called as the ethnic variations. So genetic polymorphism and personalized medicine, the process is, of the replication is not 100% perfect, and occasionally mutation can occur. If mutation does not prove fatal, it will be carried from generation to generation. This leads to different individuals having subtly different gene sequences. On average, there is a difference of one base pair in every 1,000 base pairs between individuals. It is known as the genetic polymorphism. As the nucleic acid bases act as the code for the amino acids in protein, the difference at the level at this level results in a different amino acids being introduced into the protein, which may or may not have an effect on the protein's activity or the function. Genetic polymorphism has an important consequence with the respect to the susceptibility of the individuals of the disease, also the kinds of the drug therapies that are best suited for individual. A detailed knowledge of patient's genome opens up the possibility of predicting and preventing the disease as well as choosing the ideal drug therapy for the patient should the diseases occur. This is known as the personalized medicine. So the statement which is saying that the genome mapping of the patient is not required, it is false because it is clearly stated here. I will highlight it for you that a detailed knowledge, this position, the detailed knowledge of the patient's genome opens up the possibility on the predicting and preventing the disease as well as choosing the ideal drug therapy, which is known as the personalized medicine. So this is the logic, or this is the rationale behind this personalized medicine thing, okay, which is intended to cure this genetic polymorphism, this thing, okay. So the statement number C is false, other statements are correct. Okay. Now I will go to the next problem. Problem number 14, it says that which of the following statements regarding alkaline ester hydrolysis is false? Your options are number A, this is called a saponification reaction. Number B, this reaction is bimolecular. Number C, at the first step, base attacks and form a square planar intermediate. And number D, the nucleophilic attack on carbonyl carbon is the rate determining step. Which of the following statements regarding the alkaline ester hydrolysis is false? Again, I will repeat. Number A, this is called the saponification reaction. Number B, the reaction is bimolecular. Number C, at the first step, the base attacks and forms a square planar intermediate. And number D, the nucleophilic attack on the carbonyl carbon is the rate determining step. Which of the following statements regarding alkaline hydrolysis is false? I will wait for a minute, then I will check the chat box. Meanwhile, let me know A, B, C or D, which of the following you think is the false statement regarding the alkaline ester hydrolysis. Which of the following statements regarding alkaline ester hydrolysis is false? Number A, it is called the saponification reaction. Number B, the reaction is bimolecular. Number C, at the first step, base attacks and form a square planar intermediate. Number D, the nucleophilic attack on the carbonyl carbon is the rate determining step. Okay, let me check the chat box. Let me see if there is any answers regarding the problem number 14. Okay, I got another answer. I got an answer which is number C. Let me check if number C is the correct answer or not. So the correct answer will be the statement which is false regarding the alkaline ester hydrolysis will be number C. At first step, the base attacks and forms a square planar intermediate. This is false. Okay. So what is the mechanism of the base catalyzed ester hydrolysis? So as you can see, the step one is the nucleophilic addition of the hydroxyl to the carbonyl carbon, and it forms tetrahedral geometry. Okay, intermediate. Okay, so at the first step, it forms a tetrahedral intermediate, and the second step is the elimination of OR, so which gives us the acid, which is the reproduction of the carboxylic, which gives us this particular acid, and the reproduction of the carboxylic acid also forms like this. Okay, the carboxylate ion, but the opposite thing does not happen. Okay, so the step 3 is the makes base catalyst ester hydrolysis irreversible, since the carboxylate ion can no longer be attacked by the nucleophile. So as you can see, this is feasible, but this is up to this is feasible, but the nucleophilic attack of OR minus to the carbonyl carbon is not feasible. Okay, so the first the is the so here is the we considering the alkaline hydrolysis of the ester. It is called the saponification alkaline ester hydrolysis. Now the mechanism of ester hydrolysis is basis discussed here. Suppose this is your base. Okay, and the base first attacks and forms a tetrahedral intermediate. This is the tetrahedral intermediate, and this is the rate determining. This is the rate determining step. This attack by the nucleophile on the carbonyl carbon. It is a bimolecular reaction. Two molecules are involved to form the intermediate via the transition state. The next step is the O minus comes back, and the leaving group leaves, so you get the hydrolyzed product. Okay, then the Y, suppose the Y is OH minus, then you get this carboxylic acid, and this means the hydrolysis of an acyl system ester goes to the tetrahedral intermediate via a transition state. Okay, so number C, which says that at the first step base attacks and form a square planar intermediate, it is false. It does not form a square planar intermediate, it forms a tetrahedral intermediate. Okay, so yes, very good, number C is the correct answer. Now I will go to the next problem. Problem number 15 says the enzyme which plays a major role in the biosynthesis of phenylalanine is number A isomerase, number B chlorismet mutase, number C hexokinase, and number D decarboxylase. I will be a little quick from now on because we have a couple of more questions and to discuss. So I will give you only a minute. Try to answer if you know. If you doesn't know, if you want to try, it is also fine. That's not the thing which I want to say. But the enzyme which plays a major role in the biosynthesis of phenylalanine will be number A isomerase, number B chlorismet mutase, number C hexokinase, and number D decarboxylase. Which is the correct enzyme which plays a major role in the biosynthesis of phenylalanine? Isomerase, chlorismet mutase, hexokinase, or decarboxylase? So, which enzyme plays a major role in the biosynthesis of phenylalanine? Let me check the chat box. Let me see if there is any answer for this problem number 15. Okay, I got one answer which is saying it is number B. Let me now check if number B is the correct answer or not. So, the enzyme which plays a major role in the biosynthesis of phenylalanine will be, very good, it is number B, chlorismet mutase. So, the chlorismate mutase is an enzyme that plays a crucial role in the biosynthesis of aromatic amino acids and organisms like bacteria, fungi, and the plants. Specifically, it catalyzes the rearrangement of the chlorismic acid to the prephenic acid, which is the first committed step of the synthesis of phenylalanine and tyrosine via the Shikimate pathway. So this is the first step. This is the Shikimate pathway, you know. So this is the chlorismate, and this is the chlorismate mutase converts this chlorismate to the prephenate. This is the structure of prephenate, and this prephenate is converted to the 4-hydroxyphenyl pyruvate, which gives rise to the tyrosine, and also it is converted to the phenyl pyruvate, which gives rise to the phenylalanine. Okay. Also, this chlorismate gives rise to the anstranilate, which gives us the tryptophan. So these are the aromatic amino acids basically, which are generated from this chlorismate via this chlorismate mutase enzyme. So the function chlorismate mutase facilitates the Claisen rearrangement, converting the chlorismate to prephenate. This rearrangement is only known enzymatically catalyzed by the pericyclic reaction in the primary metabolism. This process accelerates the reaction by the factor 10 to the power 6 compared to the uncatalyzed thermal process. Role of the metabolism. The Shikimate pathway is responsible for the synthesis of chlorismate, which is common intermediate in various compounds, including the aromatic amino acids like phenylalanine, tyrosine, and tryptophan, along with the other essential compound like salicylic acid, folate. Chlorismate mutase acts as a branch point of this pathway, channeling the chlorismate toward the synthesis of phenylalanine and tyrosine while diverting it away from the synthesis of tryptophan. Significance: chlorismate mutase is essential for the survival of organisms that rely on the Shikimate pathway for the aromatic amino acid biosynthesis. As animals lack this pathway, its role in regulating the balance for the aromatic amino acids within the cell is vital. Mechanism: the mechanism involves a serious step like capture of the specific conformer of chlorismate, confinement of the reaction centers, rearrangement facilitated by the transition state stabilization, ionic hydrogen bond. The determining step may not be the rearrangement itself, but rather the confinement of the reactive centers to the close distance. Regulation: the activity of chlorismate mutase can be regulated through the various mechanisms, including allosteric regulation, as you can see in the case of the yeast chlorismate mutase, which is activated by tryptophan, inhibited by tyrosine. Some chlorismate mutase, like they found in Mycobacterium tuberculosis, may even require complex formation with the other enzyme, such as DAHP synthase and optimal activity and regulation. Research interest due to its unique enzymatic mechanism involving the pericyclic reaction. Chlorismate mutase is a model enzyme for computational mechanistic studies. It is essential role in various organisms. It makes it attractive target for developing new antimicrobial agents and herbicides. Okay. So this is about the chlorismate mutase. You do not need to remember all the mechanisms. Just try to remember these few steps. Okay, the conversion of the chlorismate to prephenate and the other conversions, how it converts to the aromatic amino acids. Now let me go to the next problem. Problem number 16. It says the Sanger method of DNA sequencing is dash method for DNA sequencing. Your options are number A, an enzymatic method, number B, chain termination method, number C, D-deoxy method, and number D, chemical method. Sorry. The problem 16, it says the Sanger method of DNA sequencing is which type of the method of the DNA sequencing? Enzymatic method, chain termination method, D-deoxy method, or chemical method? Let me know in the chat box A, B, C, or D which of the following will be the correct answer. The Sanger method of DNA sequencing is known as which type of method of DNA sequencing? I will wait for a minute, then I will check the chat box. Let me check. I think it should not take much time, this type of question. Okay, I got one answer which is saying it is number D. Let me now check if number D is the correct answer. So the correct answer will be the Sanger's method of DNA sequencing is known as the number B, chain termination method. Let us discuss what is the Sanger sequencing. So Sanger sequencing is also known as the chain termination method, is a fundamental DNA sequencing technique that determines the order of 4 nucleotide bases in a DNA segment. Developed by Friedrich Sanger and his colleagues in 1977, it was his first widely used method for sequencing of the DNA, which earned the Sanger his second Nobel Prize. Despite the emergence of newer, faster, and higher throughput technologies, Sanger sequencing remains relevant in the specific application, particularly in the smaller scale projects for validating variants identified by other methods. It holds a gold standard status due to its high accuracy, typically around 99.99%. The core principle behind the Sanger sequencing relies on the selective incorporation of the chain terminating D-deoxynucleotides during the DNA replication. Here the simple breakdown, the template preparation, so that these are the steps for the Sanger sequencing. Okay, I will show you how it is done in a manner, in a pictorial manner also, and we will discuss how it is happened. So first is the template preparation. The DNA segment is to be sequenced is isolated and purified, then denatured into the single-strand DNA. Primer annealing, a short single-stranded DNA primer complementary to the known sequence is at the end of the target. It is annealed to the template. Chain termination by PCR. Chain termination PCR, the primer is elongated by the DNA polymerase in the presence of both normal deoxynucleotides and the small amount of fluorescently labeled DDNTPs for each basis. When the DDNTPs is randomly incorporated, it terminates the DNA strand because it lacks the 3-hydroxyl group necessary for the further elongation. The process creates a collection of DNA fragments on varying length, each terminated by the specific fluorescently labeled DDNTP. Size separation, the fragments are then separated by the size using capillary electrophoresis. These fragments migrate through a gel matrix or capillary with the smaller fragments moving faster than the larger one. Sequence detection, the fragments pass the laser detector for the fluorescent level are excited, the immediate light is recorded. Since each DDNP is a unique fluorescent level in order of the fluorescent signal indicates the sequence of the terminating bases, therefore the original DNA sequence. Software analyzes this signal and generates a chromatogram which is visually represents the sequence. Okay. I will show you how it is done, and I will also show you how to do the DNA sequencing by this Sanger's method. Okay. Let us move to the next question. Okay, before that, let us discuss about the differences between the Maxam-Gilbert and the Sanger sequencing. Maxam-Gilbert sequencing is based on the nucleotide-specific partial chemical modification and subsequent DNA cleavage, whereas the Sanger sequencing is a process of the selective incorporation of the chain-terminating D-deoxynucleotides by the DNA polymerase during the in vitro DNA replication. This method, Maxam-Gilbert, is developed by the Alan Maxam and Walter Gilbert in 1977 and 1980, and it is known as a chemical method for sequencing. Whereas the Friedrich Sanger and colleagues developed the Sanger sequencing in 1977, and it is known as the chain termination method. Maxam Gilbert one uses large amount of hazardous chemical including radioactive material and hydrazine, and Sanger sequencing uses less hazardous chemicals. The Maxam Gilbert sequencing highly sensitive and highly specific. The Sanger sequencing less sensitive and less specific. Okay. So I have discussed about the Maxam Gilbert. If you remember that we have discussed what is the Maxam Gilbert and how to do it in our one of the previous questions, right? So now what we will do is we will also see how to identify the particular sequence of the bases by these two methods, okay, in our upcoming questions. So the problem number 17 is the write the DNA sequence of the template DNA strand from the following diagram obtained by the Sanger method. Number A is the 5 prime A G C T T C A G T C 3 prime. Number B is 3 prime A G C T T C A G T C 5 prime. Number C is the 5 prime C C A T C G T T G A 3 prime, and number D is the 3 prime G G T A GCAACT 5 prime. This is the particular template. You have to identify the DNA sequence of the template DNA strand by the Sanger's method. Just try it. If you can answer it is fine. Otherwise, I will show you how to do it. You don't have to worry. Once I show you how to do this type of solve this type of question, you can solve any this type of problem. Try to find out what is the DNA sequence in the template DNA strand. We don't have much time left, so I will not wait for a very long time. I will wait for a minute, then I will check the chat box and I will show you how to do it. Okay. So what will the DNA sequence of the template DNA strand? From the diagram obtained by the Sanger's method. From this diagram, you have to identify what will be the sequence of the template DNA strand. Let me check the chat box if there is any answer for our problem number 17. Let me check it first. Okay, I have got one answer, number D. Very good. Let me check if number D is the correct answer or not. And I think I have got also number B. Okay, I have got two answers. Let me check which of the following is the correct answer. So the correct answer will be, it is number A, 5 prime, A, G, C, T, T, C, A, G, C, T. Now let me show you how to do it. Okay. So basically, in case of the DNA Sanger sequencing, the step one is a PCR with the radioactive deoxynucleotide. Deoxynucleotides are added to the normal PCR mixture, which includes regular nucleotide, because the addition of deoxynucleotides to an elongating DNA sequence causes premature termination of the sequence. Radio labeling of the deoxy allows to researchers to visualize the last nucleotide of the PCR product sequence. Okay. Second step is the gel electrophoresis, and third step is the decoding the sequence. So for example, this is the template you are given. At the top we have the larger fragment, and at the bottom we have the smaller fragments. Okay. So you have to remember, this is the 3' end and this is the 5' end. Okay. So from the 3' to 5' it reads as like this. We have a the sign in the T. So it is T, C, G, A, G. Then we have again T, C, A and G, 5'. Okay, so this is 3' to 5'. These are the code. Now, in case of Sanger sequencing, one important thing has happened, that you have to make it the complementary. Okay, to have that input template strand, we have to take the complementary from this, whatever the thing you have got, you have to make it the complementary one. Okay, so the complementary of this will be 5', A, G, C, T, T, C, A, G, T, C, 3', which is the answer number A. Okay. So this is the thing you have. So just remember, this is the, I will just show you. This is the 3', this is the 5'. So first you have to read from this 3' to 5' what it is showing you, and then you have to just make it complementary to have this input. Okay. So you can do it from the otherwise also. You can also read it like this: 5' G A C T G A A G C T 3'. So the complementary will be like this. So this is how the Sanger sequencing is done. Okay. Now I will go to the next problem. Problem number 18 is the same. It is asked that you have to identify what will be the template DNA from the following diagram obtained by the Maxim Gilbert. This is the diagram. You have to identify which were the particular template DNA strand DNA sequence by the Maxim Gilbert method. Number A, 5', GATG 3'. Number B, 5', CTA CGTA 3'. Number C, 3', GATGC 5'. Number D 3 prime CTA CGTA 5 prime. What will be the DNA sequence of the template DNA strand from the following diagram obtained by the Maxim-Gilbert method? I will wait for 2 minutes and then I will show you the correct answer, and I will check the chat box first, then I will show you the correct answer, and I will show you how to do the same thing in the case of the Maxim-Gilbert method. Let me wait for a minute, then I will do it, okay? Try to do it. It is very simple actually. Once I show you, you can do it easily from the next time if you cannot do it for now. Write down the DNA sequence of the template DNA strand following the diagram obtained by Maxim-Gilbert method. Let me check the chat box. Let me see if there is any answer for problem number 18 for the Maxim-Gilbert one. Do I have any answer? No. Okay, it is fine. Let me show you how to do it. It is very easy. Once I show you, you can do it easily from next time. So the correct answer will be, it is number B. Okay. So in case of Maxim-Gilbert, the thing is very simple. Okay, only what you have to do is you have to read it out from this to the this side to this side. So I have already mentioned you, like, right? 1 second. I have already mentioned you, this is the 5 prime, this is the 3 prime. So if you see that in case of this template, now here we have the different this row is different. We have A plus G, G, C and C plus T. Why? Because Maxim Gilbert, if you remember, we have the chemical, it is a chemical method which uses different chemical reagents for this specific cleavages, right? So the first lane we have the dots in both the C plus T and C, which indicates this dot is for C. So this is C. Then we have on T, then for A, then we again have for C, then from G, then only we have this for T and we have it for A. So now you have to just read it from this to this. So the difference between the Maxim, so you have this 5 prime end is this time and 3 prime is this, and you have to read it from this to this. So the difference between the Maxim Gilbert and the your Sanger method is like, what is the difference? That you, in case of the Sanger method, you have to make the complementary of the whatever you get from this diagram. You have to make the complementary. But in case of Maxim Gilbert, you don't need to make it complementary. You can read it as it is to find the template DNS. Okay. So Maxim Gilbert sequencing required a radioactive labeling at the 5 prime end of the radioactive level at the 5 prime end of the each DNA fragments, and it is the using the gamma 32p ATP and purification of DNA. The chemical treatment generates the breaks at the small portions, one or two of the 4 nucleotide bases in each of the 4 reactions. For example, the purines A plus G and designed using the formic acid and guanines and are methylated by the dimethyl sulfide and pyrimidine and hydrolyzed using the hydrogen, and the addition of salt to the hydrogen reaction inhibits the reaction of thymine. So these are I have already mentioned. The concentration of the modifying chemicals in control to introduce the average one modification per DNA molecule. Thus, a series of labeled fragment is generated from the radio labeled end. The first cut the side of the each molecule. The fragments of the 4 reactions are electrophorest side by side by denaturing the gels, and the visualize the fragment. The gel is exposed to the X-ray film auto radiography, yielding a series of the dark bands, each showing the location of the identical radio labeled DNA. From the presence and absence of the each fragment of the sequence may be inferred. Okay. Now let me go to the next problem. Problem number 19. It says the type of topoisomerase that is required for the ATP hydrolysis is number A, type 1A, number B is type 2A, number C is type 1B, and number D is type 1C. Which type of topoisomerase is required for the ATP hydrolysis? I will wait for a minute because we don't have much time and I have 2 to 3 more questions left. The type of topoisomerase which require the ATP hydrolysis is, if you know, you can let me know in the chat box what you think is the correct answer. Type 1A, type 2A, type 1B or type 1C, which of the following type of topoisomerase that is required for ATP hydrolysis? Let me check the chat box if there is any answer for problem number 19. Okay, it's fine. Let me show you the correct answer and let us discuss it. So, the type of topoisomerase which required for ATP hydrolysis will be number B, topoisomerase 2A. So, topoisomerase 1 and topoisomerase 2. So, let us first discuss about it and we will discuss about the types also. So, topoisomerase 1, I have discussed the enzymes which cut one of the two strands of the double-stranded helix and relax the strand by reanneal the strand and generate single-strand breaks. Topoisomerase 2 is the enzyme which cut the both strands of DNA helix simultaneously in order to manage the DNA entangles and supercoils, and it generates a double-strand break. Topoisomerase 1 generally occurs in prokaryotes. Topoisomerase 2 occurs in both eukaryote and prokaryotes. It does not require ATP hydrolysis, but topoisomerase 2 requires ATP hydrolysis. The subclasses are like this: type 1A, 1B, and 1C are the subclasses for topoisomerase 1, and 2A and 2B are the topoisomerase 2. Okay, so the topoisomerase which is responsible for ATP hydrolysis is topoisomerase 2A. Okay. Now, question number 20, which is the main enzyme component of Sanger sequencing? Number A, helicase, number B, DNA polymerase, number C, nuclease, and number D, guidase. Which of the following will be the main enzyme component of Sanger sequencing? Since we don't have much time left, let me just show you the answer and let me discuss it a little bit. The main enzyme component of Sanger sequencing will be DNA polymerase. Okay. So, the Sanger sequencing utilizes the DNA polymerase, a primer, and DNA nucleotides and deoxynucleotides to synthesize new DNA strands complementary to the template strand. The DDNTPs, which is the D-deoxynucleotides, lacks a hydroxyl group at 3' position, preventing the further nucleotide addition when incorporated. By including the deoxynucleotides in small amounts, the DNA synthesis reaction terminates randomly, generating DNA fragments to the various lengths. Each of the four deoxynucleotide is labeled with a different fluorescence dye. The resulting DNA fragment are separated by the size using the gel electrophoresis. A detector reads the fluorescence signal of the each fragment as it migrates to the gel, revealing the sequence of the original DNA. The read length of the Sanger sequence is typically 900 base pairs. Okay. So this is the Sanger method, synthesizes the complementary DNA strand in vitro. In each tube, we have the normal N bases, deoxy N bases, rDNA polymerase, primer, buffers, and salt. First is the preparation of the four reaction solution, followed by gel electrophoresis by autoradiography, and we have a radioactively labeled primer here. This is the DNA synthesis. Longer fragments will be here and shorter fragments will be in the less position. Now, read the sequences of the new strand and then make it complementary to have the template sequence. Okay. So, DNA polymerase is the main enzyme component of Sanger sequencing. Now, the last question of today's session: which of the following statements regarding luciferin is false? Number A, it is present in the fireflies. Number B, it reacts with the ATP to generate luciferase. Number C, it associated with the generation of lights in fireflies. Number E, none of the above. This is the statement which is false regarding luciferin. I have already discussed it when I was discussing the pyrosequencing. Is the number B, that it reacts with ATP to generate luciferase? No. Basically, the reaction of the pyrophosphate plus adenosine phosphosulfate in presence of the enzyme ATP sulfurylase give ATP plus sulfate. So, a molecule of high energy is generated which is known as ATP. Now, ATP reacts with a compound called luciferin. Luciferin is a compound which is presence in the fireflies, which gives lights. The luciferin presence in presence of molecular oxygen and an enzyme is luciferase reacts with ATP to form oxyluciferin. Okay, now, luciferase is the enzyme. The luciferin in presence of ATP with the help of the luciferase converts is the oxyluciferin. This is the structure that generates light. So in the pyrosequencing, you take your DNA strand, add your primer and requirement. Suppose you have added the DATP, then you have added ATP sulfurylase, you have added luciferin, and you will add the luciferase enzyme to generate light. Now, when luciferin reacts with ATP in presence of oxygen. Okay. So the statement which says that it reacts with ATP to generate luciferase, it is incorrect. It reacts with ATP to generate the oxyluciferin, which gives the light, and it is the light which is you can see in the fireflies. Okay. So this was the last question of today's session. Here it is also the same thing is mentioned, how this luciferase the D-luciferin is converted to the oxyluciferin in presence of this luciferase, and magnesium is also present here, which gives the light. So this was the last question of today's session. So today we have discussed about the DNA sequencing method, the Sanger's method, Maxam-Gilbert method. We have discussed about the different types of DNA and all the related stuffs. So today it was our week 5 live session, and now I will share my email ID. So this is my email ID. If you have any doubt or any query, okay, once again. So this is my email ID. If you have any doubt or any query regarding today's class or any previous class, please feel free to reach me out through the email. I will answer your queries through email also, and I will answer your queries. If you have any particular doubt, I can discuss it in the upcoming classes. So today it was our week 5 live session. I thank you all for joining and answering during the class. I will share this PPT and the particular lecture video in the SWAM portal shortly. Okay. So thank you all for joining. Let's meet on the next Friday in our week 6 live session with the same time and same Google Meet link. Okay. Thank you all. I will close the session for today here.